Hello and welcome to Science Research Weekly, Season 4, Episode 25, Double Detonation Supernovae. Get ready for the Big Five workouts, CryoRhodopsins, Double Detonation Supernovae, Moonrise, the 4 layers of testing for R packages, and a 3-track grant from the NSF on translation to practice. This episode covers materials from July 4th to July 10th. Let's get started with some research articles. First up, personality traits can predict which exercise intensities we enjoy most and the magnitude of stress reduction experience following a training program. Very descriptive title. So researchers in this study used the big 5 personality traits, OCEAN, or openness, conscientiousness, extroversion, agreeableness, and neuroticism, to predict engagement with a physical fitness program. And it turns out different personality traits were good predictors of certain features. This suggests that a physical fitness program calibrated to personality type may improve engagement. Some of the particular things that they found, things like extroversion predicted greater enjoyment of the highest intensity, conscientiousness sort of predicted overall fitness and more weekly hours. But I think the biggest thing was neuroticism. People who scored high in neuroticism benefited the most from stress-reducing effects of aerobic training, and they were more likely to stick with things when they weren't being in the lab. They were doing things on their own. They weren't being sort of observed. All right, next up, the article CryoRhodopsins, a comprehensive characterization of a group of microbial rhodopsins from cold environments. So rhodopsins are light-sensitive membrane proteins with potential for use in things like biotechnology. And there's an interesting group of rhodopsins that have been found in cold-dwelling microbes, dubbed the CryoRhodopsins. Given their ability to sense UV radiation, they might be a good target for a photo switch in a biotechnical application. Moving along here, we have the article, The Genome of a Sea Spider Corroborates a Shared Hox Cluster Motif in Arthropods with a Reduced Posterior Tagma. So, sea spiders, aka pycnogids, are members of subphylum Chelicerata, along with arachnids and horseshoe crabs. And sea spiders are pretty weird. However, figuring out their genetics has been hampered due to no high-quality genomic resources for pycnogids. Now, researchers have a complete genome and complete and high-quality genome using long-read sequencing. One big finding was that there was no evidence of a whole genome duplication, WGD, which is a trait found or an event found in other chelicerates. Next article here, regulating multifunctional oxygen vacancies for plasma-driven air-to-ammonia conversion. Ammonia, or NH3, is important as a fertilizer. It's really important, actually, as a fertilizer. And most ammonia is synthesized using the Haber-Bosch method, which is great, but it takes a lot of energy to produce. So if there's an easier way to do it, less energy intensive, be fantastic. Researchers here were able to use actually plasma, so electrified air, to combine nitrogen and oxygen from the air into NOx, pass it through a catalyzer made of, I think, iron and copper sort of nanoparticles to actually create ammonia. So this has, a real potential to speed up, simplify, reduce the cost and effort of ammonia production. Okay, think about other elements, but out in space, we have the article Calcium in a Supernova Remnant as a Fingerprint of a sub-Chandrasekhar-mass Explosion. So type 1a supernovae happen when a white dwarf accretes enough matter from another source, typically a companion star in a binary star system, and then that white dwarf with accreted matter detonates. Now, astronomers have actually imaged the remains of supernova SNR 0509-67.5, which actually may have had a double detonation instead. A double detonation is apparently where an outer envelope of helium on the star detonates first, sending a shockwave that also detonates the core of the white dwarf. The evidence for this in this observation, SNR 0509-67.5, is actually a double shell, with one shell being calcium, the other being sulfur. Okay, moving along here to our penultimate article, restoration of striatal neuroprotective pathways by kinase inhibitor treatment of Parkinson's disease linked LRRK2 mutant mice. So leucine-rich repeat kinase 2, that's LRRK2, it's a protein associated with Parkinson's disease, and it suppresses cilia formation in brain cells. However, the molecule MLI-2 might actually inhibit that LRRK2. So researchers were able to feed Parkinson's disease mouse models with MLI-2, found that the treatment restored cilia formation, suggesting that some of the effects of Parkinson's disease, if MLI-2 works in humans, could be reversed if administered soon enough. So really fascinating, promising work here on not just delaying, but restoring issues with Parkinson's disease. And then closing out our time on the articles here, a short news point about moonrice. It's called moonrice, developing the perfect crop for space bases. Talking about these researchers trying to create not just dwarf rice, but actually super dwarf rice to be able to grow in space, because size is a premium. They need to be very small and to be able to grow reliably in space, but not just small, but productive. So that's what the moonrice project is trying to do here. It's apparently a collaboration of 3 Italian universities. So with that, let's turn to some research tools. There are 2 new R-bloggers posts. First, from Jacob Sobolewski, the 4 layers of testing every R package needs. The layers are number 1, unit tests to catch bugs at the source, acceptance tests to ensure user goals are met, code coverage to highlight what you're missing, and then mutation testing, check if your tests are meaningful. Second blog is from the Jumping Rivers blog on evaluating your skills and boost your career with Jumping Rivers free monthly webinars, upcoming webinars. In August, there's reports that write themselves, automated reporting from Quartum. In September, there's building scalable Shiny apps with asynchronous programming. In October, we have understanding POSIT, ecosystem and enterprise use cases. November brings us machine learning with Python, and closing out the year on December, accessible Shiny, designing for all users. 4 new CRAN packages that might be of interest. There's BMI Select, Bayesian MI Lasso for variable selection on multiple imputed datasets. There's GGGDA, a ggplot2 extension for geometric data analysis. There's Reptile BR, interface for the Reptile database for querying and retrieving taxonomic data, and SEM Effect, structural equation modeling, effect analysis, and visualization. All right, then, we will close out our time here together with 1 grant opportunity. There is an NSF, National Science Foundation, Translation to Practice grant. This grant is aimed to advance 3 primary focus areas: accelerating technology, translation and development, fostering regional innovation and economic growth, and preparing the American workforce for future high-wage jobs in STEM fields. There's actually 3 tracks available. There's the NSF TTP Explore, a pilot track that's likely to be the first step for researchers seeking to translate their basic research to practice. There's a translate version, which is starts with use-inspired research and initial translation activities and further matures that into practice. And then there's the partner one, which supports translational efforts to demand 1 more partnerships for technology development and deployment. Double shell supernova across the sky. Thanks for joining me for Science Research Weekly. Science on.